The overall goal of the following experiment is to study the effects of spinal cord ischemia in a murine model. This is achieved by first inserting a laser Doppler probe over the femoral artery. Next, the sternotomy is performed to access the aortic arch and left subclavian artery. Then, careful dissection of the aorta and subclavian arteries is performed in order to adequately reduce arterial blood flow to the spinal cord. Results are obtained that show a postoperative neurologic deficit which is based on the Basso-Mouse score.
The main advantage of this technique over other existing methods such as the lateral thoracotomy is that the pleural space is not violated.
This method can help to answer questions for the pathophysiology of spinal cord ischemia-reperfusion injury and provide potentials for pharmacological interventions.
The implication of this technique extends towards therapies in preventing paraplegia following spinal cord ischemia.
Though this method can provide insight into paraplegia, which complicates thoracoabdominal surgery, it can also be applied to other disease processes causing acute spinal cord ischemia, such as hypotension or vasculopathies.
Generally, individuals new to this technique will struggle with the dissection of the aortic arch because extreme care is needed to avoid hemorrhage or pneumothorax.
So we first thought about this technique after we had several difficulties with lateral thoracotomy approach, which is widely published in the literature, but was associated with a higher mortality in our animals.
Visual demonstration of this method is critical, as extreme caution is needed during the mediastinal dissection to prevent intraoperative mortality.